I'll be the moderator today, and I'm actually working from home, like most people at SPEX right now. But we do have Eric Smith in the lab, and you should be able to see him now with his lab coat on, and he'll be the actual demonstrator today. This is our first live demo. We're really excited to be giving this live demo. We hope it goes smoothly, and we hope that you have a good connection. We're going to actually show a couple of our main products today. The first that we'll be showing is the GenoGrinder, which is a tissue homogenizer. And then we'll be showing the FreezerMill, which is a cryogenic grinder. So I'll be handing over to Eric in a minute or so to start the demo. Before I do that, I just wanted to give an update about SPEX. Fortunately, we are still able to manufacture here in New Jersey. So we have our full manufacturing personnel in the offices still manufacturing our products. They are ready to ship. And the rest of us are working from home, but we are all available to assist our customers with any questions or application requirements. A little bit more about SPEX. We've been around for over 65 years now. We celebrated our 65th anniversary last year. And we make a range of different sample preparation equipment for different analytical techniques. And our goal is to help you achieve accurate and consistent results by ensuring reproducible sample preparation analysis. So without further delay, I think I'll hand over to Eric. I'm going to mute my microphone, and then I'm going to make Eric the presenter. So let me just do that quickly. Okay, Eric is now the presenter. And you should be able to see Eric. If you go to the viewpoint and you can actually select who's talking, that will allow you to see the full screen of Eric. And I'm going to mute my microphone and hand over to Eric to continue with the demonstration.
Okay, thank you, Tim, for the introduction. My name is Eric Smith. I'm the application scientist at SPEX SamplePrep. Today, we're going to conduct a few demonstrations with the SPEX 2010 GenoGrinder and the 6775 FreezerMill. The GenoGrinder is a high-throughput homogenizer, often referred to as a bead beater, simply because it uses balls made of steel, ceramic, or glass to pulverize a sample. Some features of the GenoGrinder include programmable runtime, cycles, rest period, and grinding speed. Operating commands through a series of touchscreen displays, administrative lockout, self-diagnostics, a large observation window, an adjustable clamp that can accommodate tighter plates, jars, tubes, and vials. Sample types include plant material, tissue samples for nucleic acid extraction, seeds for crop science, marine biology, pesticide extraction by the catcher's method. We're going to demonstrate using a 15 milliliter short vial with 2 steel balls and homogenize corn, seeds, barley, and coffee beans in a short time. So here we have the GenoGrinder here. To access the grinding area, we want to raise the lid. To remove the adjustable clamp lid, we're going to press the quick release button and lift the lid over the threaded rod. At this point, we can add the sample holder, which has a hole notched out to fit over the threaded rod. Slide down to the clamp base. We can add in our samples, coffee, barley, and seeds. To replace the lid, we simply are going to slide the clamp down over the notched out point for the threaded rod and the gliding in the back. We want to hold the quick release button while sliding down. We want to give a couple of turns to lock and secure the vials. Then we can close the lid We're going to set the runtime for 2 minutes with a rate of 1,700 strokes per minute, a 0 rest period, and 1 cycle. Then we press run.
Okay, thanks, Eric. I'm going to actually come back online here while Eric mutes his microphone, just so that we don't hear the machine running in the background, which makes it difficult for Eric to talk. But as you can see, the GenoGrinder is now running those samples. You can visually see through the window the clamp moving up and down with the samples inside and the grinding media. And while that's happening, which is going to be going on for about 2 minutes, I thought I'd show you just one of our quick start guides that are available for each of our products. This one for the GenoGrinder has a very simple introduction to show you how to operate the controls and the clamp. And in particular, I just wanted to go in and focus on the control panel here that you couldn't quite see in detail during Eric's demonstration. So here we have the control panel, and then we have some buttons here to take you to the settings, where you can put in your runtime, your speed, which is the rate up to 1,750 RPM. And then we can actually run different cycles. So you can program in the number of cycles here, and then you can have a rest time in between each cycle. So once you've done that, you can go straight to run, or you can go to the store protocols page by hitting the store recall button here. Here you can use the keypad to program in your protocol, giving it a name either for your sample type or the protocol that you want to give it, and then you can save that for quick recall in the future. In addition, we have the resources button. So here, this is going to bring you to the resources page where we have the manual available, the full manual. We have some demonstration videos of the products and some of the accessories being used. We also have a run history, further instructions, and also a complete list of accessories. So this quick start guide is available as a PDF, and if you're interested, you can request this from your sales representative. Okay, I'll just bring Eric back into view because I think that will be almost finishing. And hopefully you can see Eric now. Eric, are you there?
Okay, I'm here, Tim. Are you ready to continue?
Yes, let's go ahead, Eric, and continue with your demonstration.
So we see that the run is complete. Simply grab the lid handle and raise the lid. Turn slightly the locking knob, press the quick release button, and pull the lid off the threaded rod. As we can see, our samples are completely homogenized. I'll show one for demonstration. What you want to do is remove the grinding balls. So we can see that the sample was nicely ground in a short period. The other samples I have before and after samples already prepared. So here are the seeds. Coffee beans, the corn, and the barley. Using the 15 milliliter short vials, we can run 12 vials simultaneously in this holder. For higher throughput, we can actually stack 2 holders and run 24 vials. The next demonstration I would like to do is with tissue sample, showing how the GenoGrinder is capable of grinding tissue sample up very simply. The technique that we're going to use for that, we're going to use actually the cryo blocks. The cryo blocks are aluminum blocks that are designed to keep the sample cold during the grinding process. They can be chilled either with liquid nitrogen, dry ice, or in a freezer, depending on how tough the sample is to grind. The cryo blocks that I prepared today actually were chilled in liquid nitrogen. Since they're cold, I want to protect myself by using cryo gloves because the blocks are very cold. Simply what we do, we place a cryo block into the clamp. Here you can see I have a meat tissue sample, 1 ball in a 50 milliliter tube. This tissue sample that I'm using today is actually chicken gizzard, which is a good example of a fibrous material that can be ground. I have 1 gram of material in each vial. When running cryo blocks, you must run 2 cryo blocks simultaneously in order to balance the clamp. Each cryo block can run 6 tubes for a total of 12. Just as the foam holder, to replace the lid, we slide the lid over the threaded ride onto the guide in the back, press the quick release button, and slide down. Give a few turns to lock in the valves to make sure everything is secure. We want to close the lid. We're going to set the run time for 1 minute and 30 seconds with a rate of 1,500 strokes per minute. 1 cycle, 0 rest. Press run.
Okay, thanks, Eric. I'll take over the mic again since we have the machine running. And again, we can see through the window here, the cryo blocks with the tubes inside clamped securely into the clamp, moving up and down at high speed. So we'll wait for this to run down, and we'll leave this on the screen for now. Heather, just a quick question for you. Are you interested in any particular samples that we're running today? What is your normal application?
I work in a pesticide residue lab, so we currently have a GenoGrinder already that we're using as part of our catcher's extraction process. So we're not necessarily grinding up samples as much as we're basically using it as a high throughput mixer, just to mix our reagents with our samples really well until we proceed with the rest of the extraction. But we are replacing an older model with this new model, so I am interested just to see what some of the differences are in running it.
Okay, thanks, Heather, for that. That's useful to know. If you could mute your microphone again so we can reduce the background noise. And I can just touch a little bit on that regarding pesticide residue extractions. We do have a large user base that is using the GenoGrinder for that purpose, for high-speed shaking of reagents with the samples for the extraction process, such as catchers. So that is a commonly used technique and very effectively done in the GenoGrinder. Looks like the run's come to a finish, so I'll mute my microphone and hand back over to Eric.
As we see, the run is complete. Just as before, we lift the lid by the handle and raise upwards. We take the locking knob to press the quick release button. We can remove the lid. So here we have a sample showing that the tissue is completely homogenized. I'll give you an up close view. Sorry, that's the ball. So here you can see that the tissue sample is completely in powder form. Now, when the sample is actually frozen and ground, it will return back to a consistency state because there's a lot of moisture content. So either the sample must be prepared to work with immediately, or it must be kept cold until you decided to take it to the next step. Okay. So this actually concludes the demonstration with the GenoGrinder, and we can move forward to the 6775 FreezerMill. Okay, so the SPEX 6775 small FreezerMill is actually one of 3 cryogenic mills that we offer. We have a large 6875 and a large 6875 dual model. The SPEX FreezerMills are capable of grinding almost any sample, such as plant material, animal tissue, hair, bone for nucleic acid extraction, seeds, crop science, plastics, pharmaceuticals, food products, circuitry boards for RoHS, and textiles. Samples are chilled in a reservoir of liquid nitrogen while simultaneously being pulverized. Our proven technology uses a dual electromagnetic grinding chamber that rapidly drives a steel impactor back and forth against 2 steel end plugs. Some features with the GenoGrinder, with the FreezerMill, which are similar to the GenoGrinder. It includes programmable functions for pre-cool, which is the time allowing the sample to reach cryogenic temperatures before being ground, runtime, cycles, cool time, which is a rest time in between the cycles, and the impactor rate, and operating commands through a series of touchscreens. The operation is simple. Micro vials, small vials, mid-sized vials, and large vials are added to the grinding chamber to ground the sample. I must note for the 6775, only the micro vials and the small vial will fit into this mill. The other 2 models, larger mills, can run every vial available, from large, mid-size, small to micro. One thing I must also mention that it is important to observe safety when handling liquid nitrogen. Chilled vials, cold surfaces in the FreezerMill, proper protection, PPE, such as gloves, lab coat, safety glasses, or even a face shield must be worn. With respect to time, we're not going to actually run the FreezerMill because it needs to go through a pre-cooling cycle, but I'm going to demonstrate how to add the sample and remove it, and then show before and after samples. So to utilize the mill is latched down, touchscreen is on top of the mill. You simply pull the latch to raise the lid up. Inside the mill, we have the grinding chamber and also other stations that pre-cool samples. The tub of liquid nitrogen is here where the sample is immersed and then ground to a finer size. So wearing my cryogenic gloves, you want to take the sample. Here I have a sample of a cotton ball, which can be ground to a powder form and possibly analyzed for viruses, including the coronavirus. The sample is placed in the vial alongside of the impactor rod. You want to take the vial, place it inside the grinding chamber, slowly close the lid to avoid splashing of liquid nitrogen. Latch the lid shut and program the cycles as needed for the sample. When it comes time to removing the lid, with your cryogenic gloves, unlatch the lid. Raise the lid up. I have a pre-ground sample here. Take the sample out. You can use the extractor. To place the vial in and remove the end plug. Next, you remove the impactor out of the vial, and now we can dump the vial. Show the contents. So here, we can see that the cotton ball was reduced to powder form. Some of the samples that we can demonstrate to also show how the FreezerMill can easily grind samples. We have here a cotton applicator tip. That can also be analyzed for possible swabs for virus. And also we have a laboratory glove, which is a fingertip that was cut off, placed in a FreezerMill, and cryogenically ground. And we can see that the sample is completely homogenized, which can be used for the next step in testing. So this kind of includes what I had to offer today, Tim. I'm not sure if you want to add anything else.
Hi, Eric. Yeah, I'll take over from here. Thanks very much for the demo. That was fantastic. So if you could mute your mic, and then I'll carry on talking here. This concludes our demonstration today. I hope everybody who participated found it useful. We do have a great demo program that is available if people do want to try out our products. We can ship them to you, and then you can try them out free of charge for a couple of weeks. You just have to pay for the shipping. We also have our free sample grind program. So you can send samples to us, and then we can test grind them for you and then send back the results. So we do have those two programs available for you. I just also wanted to mention that we do have a new product available right now that is in pretty big demand. We have some hand sanitizer that is now available from SPEX. So please go to our website for further information about that. And if you do have any further questions, please contact your sales rep and they'll be more than happy to help you. So thanks again for joining us today, and enjoy the rest of the week, and goodbye from us.